Hello, this is Shashank Biswas, and I am doing a project on DNA nanostructures review. This is a review paper. I am doing this paper under Professor Manish K. Gupta in the course IT 468 Natural Computing at DICT. Introduction. Nadine Seaman was the first to pursue the field of DNA nanotechnology and perform successful experiments to come up with an entirely revolutionary field of technology which could open a never-ending field of applications. With the never-ending need for products in the current world to fulfill the demands and requirements of the ever-growing population, the scientific community need a revolution to give the world something which could provide the vast needs in a very small resource utilization. DNA could be the key to that. With the current level of developments in the DNA nanostructure advancements, the time could be near when every technology could be shrunk to a level that could be carried in hand, let it be an electronic, mechanical, or biological device. In this paper, we try to address the developments that took place in the past decade and a half in the field of DNA nanostructures. Siemens' original motivation was to create a 3D lattice for his crystallography studies. He got motivation from the woodcut depth model. Nanodesigning can be approached in two ways. First, top-down approach. The top-down approach employs natural and man-made blocks that are cast, cut or machine processed into precise products, small or large ranging from circuits to jumbo jets. The methodologies to produce nanoparticles from large material require high-energy ball milling, solution chemistry, electro-explosion, sonication, mechanochemical processing, etching, and physical and chemical vapor deposition. The pattern structures that are grown by appropriate lithographic and ion implantation are cornerstone of silicon integrated chip technologies. Secondly, bottom-up approach. The bottom-up approach is manufacturing nanomaterial by changing and organizing individual atoms and molecules into specific configurations like processes performed by molecular machines in living organisms. The self-assembly lipid molecules into nanostructures like liposomes could be considered as classical demonstration of the bottom-up approach of constructing nanomaterials. One more attractive example of atom-by-atom assembly of nanostructures is the writing of the IBM logo by placing individual xenon atoms on a surface with an STM tip. Now, structural DNA nanotechnology from design to applications. It became possible to create these nanostructures by the help of two following properties: tile-based method and DNA origami method. Tile-based method is comparatively old and DNA origami method is new and more appropriate for construction of bigger and sophisticated structures. Nadine Seaman proposed the idea of DNA nanostructures in 1980s with the help of different arrangements. He created many complex DNA structures. He used arrangements like double crossover motif DX tiles, two-dimensional lattices, triple crossovers, and 4x4 cross tiles. Now, applications of DNA nanostructures. DNA nanostructures serving as a platform for directed assembly of synthetic vaccines. For the targeted delivery of antigen to the antigen-presenting cells (APC), which also include macrophages, dendritic cells, and B cells. This is an important step in starting a good immune response. Earlier researches showed the size and shape, surface charge, also hydrophobicity, hydrophilicity, and receptor interactions by an antigen can affect its adoption by APC. Then the targets are processed and offered to T cells for activation. It is seen that co-localization of antigens and adjuvants inside the same APCs can amplify antigen presentation and T cell activation. At last, activated cells assist in differentiation of antigen-specific B cells and creation of antibodies which are specific to the target antigen. Now, DNA nanostructures as templates to assemble organic and inorganic material on them. On the left, organic material templated on DNA nanostructures, and on the right, inorganic material templated on DNA nanostructures. The common strategy for making a DNA protein conjugate pair is functionalizing DNA with biotin and binding it to streptavidin. The 4x4 cross tile is modified with biotin at the center of the tile, with streptavidin is added to the mixture and bound. Clear two-dimensional periodic streptavidin arrays are seen by AFM. In later developments, streptavidin was attached on linear TX tiles arrays using